Okay, so it's 12:30, so I'm going to introduce now again Dr. David Israel, who is the Vice President and Lead Discovery Lead Discovery Sciences at HitGen, where he oversees use and advancement of HitGen's DNA Encoded Library platform. David also was an active member of the organizing committee of this workshop. So thank you, David. Welcome, and please go ahead and start your session.
Okay, thank you, Serene. So welcome back, everyone. And so yesterday we heard some really nice talks about the design and synthesis of DEL libraries from Christopher and Yoon. And then following those presentations on how the DEL libraries are made, we heard from Tracy Chen from HitGen on preparation and design of the target proteins that are used in the DEL screens, and then the analysis that's done in pre-selection experiments to be sure that those proteins are selection ready. Following Tracy's talk, we heard from Svetlana Belyanskaya on the actual selection campaigns, that is putting those DEL libraries together with those target proteins and bringing them through the selection process to come out with a population of enriched molecules that hopefully contain the desired hits from the DEL selections. Now, it was said yesterday that DEL library chemistry is the most important aspect of DEL technology. And I'm not going to get judgmental on what are the more or less important parts of DEL technology, but I will point out that 98% of a DEL molecule is DNA. The D in DEL is DNA. And there are really many good reasons why 98% of a DEL molecule is DNA. It mainly enables us to use all the powerful tools of molecular biology to both synthesize the libraries and analyze the libraries, and including but not limited to next-gen sequencing. Another critical reason that the DNA is so important is the incredible information content that's contained in that encoding DNA. And the last talk of today's, of the selections session will really focus on analyzing the DEL selection process from a molecular biology perspective and taking advantage of those tools and the information content that's contained in the DNA of the libraries. And so that last talk of the selection session will be given by Letian Kuai. Letian received his Ph.D. from the University of Rochester in upstate New York, then moved on to the Broad Institute to do a postdoctoral, his postdoctoral career, and then joined GSK in 2010, where he started working on the DEL platform at GSK. In 2018, Letian joined WuXi AppTec, where he currently serves as head of science and strategy for the HitS-RSD group. And so with no further ado, I'd like to introduce Letian to talk about the downstream molecular biology of DEL selections. Letian.
All right, thank you, David, for the introduction. I also wanted to thank the organizing committee for inviting me to give this presentation. I feel honored, and also, the task I was given was a very exciting one. I would like to echo David's comment that as a biologist, I refuse to think DEL is just a chemistry matter or even the DEL molecule itself, and the DNA part is extremely important. And second echo I wanted to make is the opening speech about the assay guide manual. I think the DEL has been around for 30 years, and starting from the initial success, more and more players are into the field, and then we all made changes, improvements here and there. And I personally think it is a time for us to to gather together and compare notes and to try to come up with a better standard for this wonderful technology. So here we go. Let me try. Do you see my screen at all, or
Not yet.
not yet? Okay, let me share my screen.
There you go.
How about now?
Perfect.
All right, so so I will be covering the next step from the selection. So mostly the qPCR, PCR, and sequencing. So as you all know, the success of a DEL is heavily dependent on the development of a sequencing technology. As you can see here, I have a rough correlation with the development of the sequencing world. The cost of the sequencing keeps going down, and the sequencing capacity keeps going up. And interestingly, there seemed to be a rough correlation with the annual paper that was published in DEL technology. And I actually share the views that I heard yesterday that, you know, to have more and more successful DEL, you need more and more sequences, and also good sequences. So, so like I said, the DEL technology has been progressed for 30 years now, and then there are many different variants of the DEL. You know, in the interest of time, I cannot cover all of them, so I will pick the most common theme and some of the general practices. And when it comes to the DEL, there are different formats. I think it's fair to say nowadays the widely used DEL is a type of DEL that is DNA recorded library. As Matt said in yesterday's happy hour, this is a brutal force to encode small molecule with a DNA. But nowadays it's no longer that brutal, and I think we can make libraries in a couple of weeks with the help of all the automation. So the DNA recorded library synthesis is a tried and true modular design of a DEL system, and it has the highest tolerance of a reaction condition, and it has a lot of advantages, and you know, allowing the chemist to explore all kinds of different chemistry. And also, you know, the modular design you know, allow us to make the library and save it in the freezer, and then we will add on the other DNA sequences later to fit for different purposes. As you all know, the DEL selection is affinity selection, and after the selection, the selected molecule needs to go through PCR and purification, and then sent to next-gen sequencing. And after that, we will be reading the DNA sequences, translate them into chemical information. So today I will be mainly covering the PCR and NGS. So as Svalana already set the stage yesterday, and almost nobody possesses only one DEL. When the DEL library is made, it is always that a library tag is added to encode the library itself. And also, it is a common practice to add on another so-called pool tag to encode the entire library collection. Okay, so as you can see, the DEL, some people ask me what is the difference of a DEL compared to the traditional combinatorial chemistry, and my answer is always being the DEL is always combine, combine, and combine, and the split and pool concept has been used over and over again, not only in the library production, but also in the selection process, sequencing process. So here is the first instance, another combination concept here that we can combine. We will put the library tag onto these libraries, and they can be stored separately, but when the library is needed, we can take them out, pull them together, and put on a pool tag. And then we will go to the selection. And obviously, this is just a very, very basic arrangement of the DNA, the add-on DNA sequences, and there is more. Okay, so once we prepare the library pools, we will send the library to the selection. There are several consideration points. The first is the round of a selection. I think it was discussed yesterday that we will try to to run more rounds of selection because we believe that it will increase the signal to noise ratio. But at the same time, the counterforce is that since the DNA recorded library is not reproducible, the more round means the diminishing of the signal strength itself. So in our practice, we found that 2 to 3 rounds seem to be the sweet spot. And obviously that is dependent on the library size and the current sequencing capacity. Another point to consider is contamination. You've already seen Svana showing the molecular number progression from the first round to the last round, and each round you have a drop of 100-fold to 1,000-fold. So imagine if you have a previous round, a tiny little droplet go to the next round, you will have a catastrophe of contamination. For that purpose, the pool tag serves as your first defense line for these contaminations. And when it happens, you have an opportunity to separate out what should be the sequence according to what should be the sample according to the experimental design. And step 3, we need to take the selected sample to PCR. And there are also a couple of things to be considered. The PCR cycles. Obviously, you run more PCR cycles, you get more PCR product, and that usually translate to better sequencing quality. However, the counterforce here is obviously the more PCR cycles, you will generate more mutants. And also the PCR input plays a role in here. If you put more selection output to serve as a PCR input, you have a more complete sampling of the selection output. However, that would obviously require you to have a much higher sequencing bandwidth. I want you to keep in mind that the current state of art sequencing has about 5 E8 to 1 E9 reads per flow cell on average. So if you have a deep pocket, you can sequence your sample with the entire flow cell. But if you want to save money, you actually have some room of combining your several samples into one flow cell. There are some allowance here, but you try not to send 100 samples per flow cell. So because of these force and counterforce, it obviously requires to make a rational decision of when to stop our selection and how much sample do we put into the PCR, and that obviously require a precise quantitation to make those decisions during experiment. Okay. And after the PCR, obviously the PCR product needs to be purified. It is in our experience, if you don't purify those leftover primer dimers, will also interfere with the sequencing and also occupy the clusters on the flow cell. Right. And the sequencing depths, that was actually a hot discussion area or even debated area. Again, the deep sampling usually means a better coverage of your sample, but also that needs to use more sequencing bandwidth. When it comes to actual next-gen sequencing, it is very standard. We have been very good at turning DEL sequences to a sequence that is adaptable to modern sequencing techniques, and there's actually not a lot of variation there. Right? The only differences are, some people use single-end sequencing. Some people use double-end sequencing, and in terms of installing the sequencing adapter, some people use ligation, some people use PCR. But those are, in my mind, trivial variants. And one rule of thumb, and everybody needs to keep in mind, is the sequencing follows a binomial rules, and that requires you to sequence 4 times of your expected input. This will give you about 100 or 90 or precisely 98% coverage. So after we get the sequences from the sequencer, obviously there will be data cleanup. And I want to highlight, what is the data flow here. The first, you need to get rid of the sequences that is not related to DEL, and there are phi-X filler into these samples. By the way, the reason of that is that DEL structure is very patent, and the sequencer don't like at any position have the same nucleotide. For that reason, a lot of applications uses mixing phi-X, to break that pattern, or you can intentionally make a frame shift on the DEL sample to avoid a patterned sequence. So if you use phi-X, obviously you need to get rid of them. And also we have observed there are always a little bit of a human genome sequences showing up in the NGS output. Obviously that is the tiny little contamination that nobody could avoid. After you get rid of the non-DEL sequences, and then you can start parsing these sequences into different things. Like I said, there are sample tags, so that you can use a sample tag to separate your different sample if you choose to sequence multiple samples in the flow cell. And library ID will help you to further separate the data into different libraries. And then you can start translate your small molecule information by the coding region. And lastly, there is actually an important segment of the DNA I haven't talked about, known as the UMI, or degenerate region. This is actually in my opinion, the most important part of in the DEL sequencing. But so far, hopefully I have described a general theme that once a DNA is made, the DEL is made, you typically will put on the library ID, and you will put on a pool ID so that you can differentiate different batches of library pools, and UMI, and obviously the sequencing adapter. Okay, there are actually different implementation of this, and there are additional accessory sequences people put into these sequences, such as anchoring sequence, so that you fight against frame shift, and also other sequences that you can use to detect mutations in the PCR. So again, the sample ID is something that is post-selection that we put into the adapter that actually provides a second layer fighting against contamination. And the sample ID could be put in both reverse primer or the forward primer, depending on what kind of a sequencing strategy do you use. Do you use a single end or dual end? And the sample index will be read by NGS, and that is the first parsing point. Okay, so this index actually allows a DEL sample to be mixed, and also with other non-DEL sample during sequencing. This is very useful. If you are not using your own sequencer, you use a commercial sequencer. Sometimes your sample will have to be mixed with other people. But this actually gives you a great advantage, to be able to work with all kinds of sequencing providers. And the pool ID is something that we added to the library after its production and before the selection. I already explained what it is. I just wanted to add here that the pool tag is great. It can not only provide a means to fight against contamination, but also, if you utilize it in a smart way, you can actually design complex experimental designs, and you can almost mimic a pulse-chase experiment, just like in biochemistry. You can use cold and hot material to figure out kinetics, and so on and so forth. All right, so the library ID, it can be added after the library production is done, or it can be added before you add your first codon for the cycle 1 building block. Okay. And now we come to the most important part, the UMI. Svetlana already explained the UMI. It's also known as a degenerate region. The aim is to make every single library molecule a unique sequence. And the UMI, typically it was added when the library is closed. That's before selection. I have seen other applications, although very rare, and people add on UMI after the selection is done through ligation. But the mainstream usually would put the UMI directly onto the library before selection. So why is UMI important? If you imagine a scenario that you have a native library, this is a oversimplified example here. If you have a naive library and you have 3 members, each of them had 3 copies, and after selection, you get 3 copy, 2 copy, and 1 copy. And then if you run the PCR, and then you will have multiple copies of each one of them, and there could be some PCR biases that will change the rank order of these molecules. And obviously, some of them were the original molecule, and some of them were produced by PCR. If this fact is uncorrected, you are likely to get a wrong impression of which molecule enriched the most. Therefore, the UMI is introduced such that every single library member is a unique sequence. And this approach is also widely used in the genome sequencing. And so in DEL, I have seen people use different lengths of UMI, try to cover the most probable building block split size. Let's see a range from 6 to 12. So there are some comments about this. So if you are using a very, very small library, and then I would say, using UMI sometimes could hurt you if your UMI does not have the capacity to cover all the molecule copies into a selection. In other words, you still might have 2 molecules share the same sequence. And that if you use a UMI approach, try to normalize that, you're going to lose your signal. And for extremely big libraries, the UMI can seem to be less useful because usually after the sequencing, your your sequence is very, very separate. So, but still, it doesn't hurt to add UMI into your library for very, very big libraries. But the UMI is not everything, and then UMI has its own limitations because the PCR and sequencer they can introduce mutants. And showing here is one of the examples. In this case, we give the PCR 3 million of input, and after PCR, and from the sequencer, we read 34 million of total reads. And if you apply if you compare that to a theoretical binomial distribution, you can see the green distribution curve here, that the main body of the curve somewhat agrees with the binomial distribution. That's the theoretical distribution. And first, you will notice there are some dispersed here. This is normal because when you order a UMI, the random sequence from a vendor, and nobody can guarantee a true even distribution of all the sequences. There always a slight bias towards A's or T's and so on and so forth. Okay. But there is another population, as you can see, very profound in this example, that they have low copies, but it's a big population. Okay, if you simply use a uncorrected aggregation method, these will become each species of this will be counted as a unique sequence. But the distribution, as you can see here, is actually forms almost a different population than the major distribution. And our understanding of this is that these are the PCR mutants. Okay, so how harmful is that? As you can see here, 10% of the input or amount is actually due to the mutant. In other words, these events didn't happen in the selection. These events were generated by PCR, also known as a ghost signal. Okay, so when this happens, your only hope is this ghost signal is randomly distributed among your data set, and they do not change your data pattern. But obviously, we don't want to bet on that because it could be mixed with other PCR biases. So there is a way to actually reduce this mutant, and in this paper published in 2017 that summarizes a lot of UMI tools that deals with this PCR mutant. So this collection of tools are also adapted by Illumina, and these tools are provided with the sequencing services. And that there are different algorithms. The concept of these algorithms are try to compare the sequence similarity and put them into different cluster and judge by the distance of these clusters, and also the relative copy counts to each other, and try to formulate a decision that there is one molecule that is actually a daughter molecule of another one. For example, here you have an ACAT that has 72 copies, and then you have an ACAG that only has 1 copy, and then there is 1 base difference. So according to the algorithm, it tends to call it a same molecule. So instead of so in uncorrected, in this example, you see 6 molecules, but if you use adjacency clustering, you would have counted 3 molecules, and or if you use a directional algorithm, you would actually count 2 molecules. And in our practice, we actually found that the directional seemed to work the best, slightly better than the adjacency. And one thing I wanted to say is so these correction tools is based on the assumption that that all the random sequences are completely random. So if you see a big dispersion here, so this is fine, but if you see a much flatter distribution, you probably need to go back to your vendor and ask for a refund, okay? Because that would also impair the function of UMI tools. So we also compared the effectiveness of UMI in relation to the sequencing depths, because as you can see, the UMI relies on the copy count difference or the outlier, how much outlier, how low is outlier compared to the general population. So if your sequencing depth is too shallow, then all you get is 1 copies and 2 copies, and then UMI will not work well. So in our test, it looks like at 1.5 sequencing depths, your UMI almost doesn't work. So the low copy outlier, very little of them get corrected. And when you increase your sequencing depths, you can see more and more those low copy outliers being corrected. At 15, it almost completely go away. So this seemed to be consistent with what people found in the genome sequencing, that there is a correlation in PCR cycles in terms of the mutation rate, and then the effectiveness of the UMI correction function seemed to be related to the sequencing depths. However, that poses a big challenge for Dell because we are thirsty for sequencing space. Okay, the more sequencing depths we give to a sample, the less useful sequence we're going to get out of that. So in practice, the recommendation, and at least from us, is to set the sequencing depths to 5 to 15, such that you have a balance between the total signal yield and the proper control of the ghost signal. And also, try not to run PCR more than 25 cycles. Okay, it is in our house rule is if you had to run the PCR over 25 cycles, you might as well not sequence it. Okay. So hopefully you're seeing so far that after the entire selection and the post-selection process is a sampling process, and I listed the summary here that from the library to the selection input, there is you can assume that is unbiased random sampling, and the only biased sampling occurs during the selection. And after that, the PCR and NGS, there is minimum to no loss of diversity or change in distribution. Okay, this seemed to be a very reliable system. That actually is very important for you to have a highly fidel readout of your selection outcome. And lastly, I wanted to comment on the sequencing coverage considerations. I know there are discussions on what is the best practices. How much should we sequence from the sequencing from the selection output? There seemed to be a consensus of at least 10% minimum, but for different reasons. And some folks believe that you needed 10% to keep your enrichment level consistent. But for us, we believe that 10% actually is the calling point. That if you have if you are 10 times more than your sweet spot, you might as well run another round of selection. Okay, so in our practice, we usually recommend selection to the point that you have 1E7 molecules per sample, and you can mix 10 samples together, give it a sequencer, and the sequence will spit out 5E8 molecules. And divided by 4, that's your minimum sequencing depths. and you will get a Fidel readout of a 1E7 per sample. And occasionally, some of our users, they'll light and they'll open, report that they have a high selection output, say 5E8. What to do at this point? We always recommend you can try to run another round of selection to bring it down to 1E7. The choice number 2 is obviously PCR everything and give it to the sequencer, also assign 5E7 sequencing space. But in this case, you will get near 5E7 back, but most of them will have 1 copies or 2 copies. And this is where your UMI tool will not work. Okay, so personally, I prefer the third solution combined with 1, which is you take out a 1E7 and PCR it up, and then provide 5E7 sequencing space. It will guarantee you to have 1E7 readout, and you will have effective UMI treatment. And at the same time, you still have the unused sample, and then you can try to run another round, try to bring that number down to 1E7 and sequence again. So as you can see, there are some decision making here. Not every single selection comes out with a consistent result. And finally, I hope that you can see after 20 years of development work, the Bell has matured to be something like truly like a barcode that you can read it, and then you can generate high quality data in general. And lastly, I wanted to give a little spoiler because WuXi runs a Dell Open and Dell Lite platform, and we have hundreds of kits sent to our users, and then they're conducting their own selections. And some of them do not even have their PCR and qPCR. And we actually is working on a system so that people can quantify their selection sample even without a PCR system. So this is something like this. It is based on a test strip, and on the strip you can after the selection you can tell how many molecules did you get? Do you have a contamination? And that will help users to make decisions when to submit a sample for sequencing. And this will hopefully come online in Q4 of this year. So stay tuned to the announcement of the 4th generation of Dell kit. Right, I'm going to end my talk here, and happy to answer any questions.
Okay, thank you very much, Letian, for that very informative and clear talk. We have a number of questions that have come in, and so I'll just read them starting at the top. So Letian, what do you view as the minimum sequence coverage for Dell selection samples? I think you in part addressed this already, but if you want to expand, here's your opportunity.
Right. I think the sequencing coverage has been used in different contexts, and there are 2 parts of it. One is how much sample do you take out of your selection output, and how much sample you give to the sequencer. As I just explained, if you give it to the sequencer, you better have high sequencing depths, so which naturally lead to 100% readout. But it's okay to take a portion of your selection outcome and send it to sequencer. In that regard, I would recommend to take out no less than 10%. Okay, if your selection output is way above your sweet spot, then I would recommend doing another round of selection.
Okay, another question. What size range of DNA molecules are typically used in Dell libraries?
What size? I'm sorry.
What size range of DNA molecules typically used in DEL library? So I guess the question is.
Yeah, right. So the full length of the DEL molecule is around 100 base or 100 base pair.
Okay. Hi, Letian. Your high sequencing depth recommendation requires you drive selections down to low numbers, for example, 1E7. This runs the risk of over-selecting to create a sample that is not usable. How do you approach getting this number right?
So I think yesterday, maybe Svalana already commented on that, depending on the protein. Some protein will have slightly higher retention, and some protein have low DNA retention. So you probably will not know what number you get after the selection. So you'll always be prepared to sequence your round 2 or round 3. In some cases, prepared to sequence your both round 3 and round 4. So when it comes to low number E7, that is actually not very low, and so this number actually is back calculated from the capacity on the sequencer, right? And obviously at different era, that number changes, and 1E7 seemed to be a kind of a reasonable total reads I can get out of a flow cell, assuming we put 10 samples in there. And at this level, again, it depends on the library size. Okay, so if your library is too big, say 800 billion, then I would say yes, maybe 1E7 is a little low. But if your library is in tens of billions, 1E7 is just right. I wouldn't go any lower than that.
Okay. There are a number of other questions. I'll read one more and then hand it back so we stay on time. And then Letian, if you could answer the rest after by typing, that would be great.
Sure.
So do you ever find that selection samples are difficult to PCR? For example, PCR inhibitors, how do you approach this?
Right. Sometimes the selection buffer could make it difficult for PCR, and one well-known case is a PBS buffer. The phosphate is inhibitory of PCR. So what we typically do is after the selection, we will run a sample cleanup, and basically redissolve the DNA into water. That seem to deal with a lot of buffer variation, and you're right. Sometimes the selection sample is difficult to PCR, but sometimes maybe you just have too low a number. That happens too. Say you have an incorrect measurement in the qPCR, you get a impression that you have a lot of molecule, but you actually don't. That is obviously user error.
Okay, thank you very much, Letian. And if you can offline type in answers to the questions that you haven't addressed in this forum, that would be great. Thank you to the other speakers at the selections session, and I'll hand it back over now for the next session.
Thank you.
Thank you very much, David.